Hi, everybody. This is Stacy Kimbrell, and we're here with the Monday Night Ignite calls. And tonight we have, starting off with the health portion of the call, is Doug Corrigan, Dr. Doug Corrigan, excuse me. And I'm just going to give him a plug right now. He has a great website with full of information. I just bought all of his information that you can purchase with all his previous recordings and his PowerPoints and handouts and whatnot. It's just a plethora of information. And if you're into the science or you have family or team members that's into the science, especially, they will love him and appreciate him. But yeah, it's still understandable for people who don't understand the complete science. So it's good for lay people as well. And it's just fabulous information. So Doug tonight is going to be teaching how do essential oils work and the science behind the bottles. And he can tell more about himself, but he explains why the molecules found in the essential oils are powerful and therapeutic. Doug has a PhD in biochemistry, molecular biology, and a master's and bachelor's in physics. And so he's going to bring so much great information to us. And he is Team YL, and he just loves sharing the information. So we're so thankful to have you with us tonight. And go ahead, Doug.
Thank you, Ignite team. Thank you for having me this evening. Hopefully everyone can hear me okay. Is that the case? My internet connection is good and everyone can see. Okay, great. So my name is Doug Corrigan. Thank you for having me this evening. Thank you for inviting me into your group to teach tonight. And hopefully I can bring some knowledge and some understanding to you to help you understand why essential oils work so well. And I'm going to hopefully remain compliant in all of my discussion. That's always a challenge to make sure you're doing that, but I always try to strive for that. So anyway, a little bit about my background. I started out as a physicist working for NASA and working for Oak Ridge National Lab. Did a lot of work on microgravity missions that flew aboard the space shuttle. I decided to go back to school and get my PhD in biochemistry and molecular biology because I wanted to get into the life sciences more than physics. So I did that. And once I graduated, I started a biotechnology company. And what we did is we engineered and developed, invented new tools to help the pharmaceutical industry discover drugs faster. So I'll go into some of this evening and how this works, but pharmaceutical companies, the way they make drugs is they screen millions of different compounds in their library, hoping to find to hit a specific target. So we develop tools to help them find those hits quicker and faster, more accurately, less false positives, less false negatives, that kind of thing. And I worked a lot with viruses and drug-resistant viruses, particularly HIV. In our work. So I was really brought up and involved in the Western pharmaceutical approach to medicine. And so when my wife, Amy, who some of you may know, started using essential oils in our home, I wasn't against them at all. I just thought that most of the effects that people had with them were basically sort of like a placebo effect. And so that was my understanding or my belief in it, because all through my career and my education, those types of natural products are not taught. They're not understood. They're not even discussed in class for one day. And so then when you get out into the real world doing this type of research, they're not discussed either. So unless you're in those types of circles, it's just not mainstream. So when Amy, my wife, started using them in the home, I wasn't, like I said, against them. I just didn't see that they were really powerful. But there were some cases in our family with our children, our two daughters in particular, who the oils really worked on that Western medicine didn't have a solution for. And then there was an instance where I had an issue where the essential oils worked very quickly. And so when I saw these things happen and I realized it was not a placebo effect because I couldn't explain it with a placebo, I started to do a lot of research on the oils themselves and the molecules, getting into the literature, looking at what research had been done. And once I understood what was happening and how they worked, and became a believer in them, then I started putting courses together to help other people understand in a simple way how they actually work and why they're so powerful. So some of that I'm going to share with you this evening in terms of how they actually function once they get in your body. And so I'm going to try to keep the discussion conceptual and not get into a lot of science that may be unfamiliar to some people. So I try to keep my discussions on a level that everyone can understand. So if I don't, please just ask a question or let me know. So I'm going to go ahead and share my screen and get right into it. All right. And I'm going to make this larger. All right. Is this visible? All right, good. So this, what I'm teaching tonight, is actually part of my 7-module series. I have a 7-module series that gives you all the basics of essential oils. This is module number 2, basically. And so this is has 7 different videos. All of the slides come with it. Okay, so this is the 7-module series. We're going into module 2. You can find all of my classes, my books, my recipes, and all of that at starfishsense.com. Tonight, if you order any 2 things or more, you'll get 30% off your entire order. Okay. I also have a book called How Do Essential Oils Work? So some of you may not like the digital classes. You might like a physical book. A lot of what I'm saying tonight and more is in this book. Okay. I've also written another book that you may be interested in that goes over called The Author of Light. It's also on the website. And here I go into how you can know more about God if you study science. So this is something I've studied, all different branches of science, and every branch of science I looked in, I found God. And so I kind of summarized some of that in this book, The Author of Light. You can get all of my classes, like Stacy was saying, on a memory stick like this, which I mail to you. And it has all my classes, all my videos. There's like 15 classes. A lot of the classes have multiple modules, like the vitamin series has 15 different videos. And so a lot of these classes are broken down into multiple modules. And then I have recipes and a frequently asked question database on here. And you can get all of that at starfishsense.com as well. If you were actually to buy all of these classes individually, it would be more expensive, like twice as much as just getting the memory stick. Now, if you order the memory stick and you rather have a link, a digital link where you can just go download it from the web, from the internet, let me know. Just put a note in there and I'll send you the link instead if you don't want the USB drive mailed to you. Okay, so let's start. So I'm going to go over a number of different facts to try to help you understand how essential oils work. All right, so I'm going to go through a number of facts. The first fact is the size of the molecules themselves. So essential oil molecules are very, very small on a relative scale, right? Size is always relative in terms of what you're comparing it to. But when you compare the size of the molecules themselves, which we call constituents, in an essential oil, they are incredibly small, okay? And they're about the same size as most pharmaceutical drugs that are designed and developed, and I'll show you that here. So here is to give you an understanding, okay? It's about 50 nanometers. All right, so somehow it disconnected. Can everyone hear me now?
I'm glad you're back because I wasn't sure how I was going to follow that. Okay.
Okay, so I don't know why that happened. Let me get back. All right. Is that good? Okay, so the size of the essential oil molecules are very small. So to give you an understanding, this is a virus particle called the West Nile virus. Most viruses are on this order of this size, 50 nanometers, which is 50 billionths of a meter. Okay. Now this virus is actually much, much smaller than an actual cell in our body. A human cell is about 1,000 times bigger than this. So the average size of the human cell is 50,000 nanometers. This virus particle is 50 nanometers. Now an antibody is about 5 nanometers, so that's even smaller than a virus. And then here on the right, this little black dot is about the size of an essential oil molecule, which is about 0.8 nanometers. Now, again, these are about the same size as a pharmaceutical drug. Why is that important? Because of the way the molecules or the constituents work, they have to be able to get inside your cell, and they have to be able to interact with the different proteins that are made by your cell. And so this very small size allows them to fit into all of the nooks and crannies of the proteins that they interact with in your cell. Okay, so that's the first thing to realize is their size. Now this right here is sort of a molecular diagram of some examples of molecules that are found in a pharmaceutical company's drug discovery library, which I was talking about earlier. They screen millions of compounds, and they all have these different molecular configurations, and they're about this size, okay? Now, if you look at the molecules found in essential oils, which I represent here on this slide, you can see they're very similar in nature, but somewhat different, but they're about the same size, okay? So these are constituents. All right, so the first fact was that their size is very small, allowing them to interact with the protein targets in our cells. The second fact, fact number 2, is that the molecules themselves are what they call lipophilic, meaning they love lipids. So they rather interact with lipids more than they would rather interact with water. You know, water and oils tend to separate and they don't interact with each other. Inside a cell, the cell is actually surrounded by a lipid membrane, or called a phospholipid bilayer membrane. That's the outside of the cell. The outside of your cells are made out of lipid membranes. The inside of your cell, where the nucleus is, where the DNA resides, is also made of a lipid membrane. And then you've heard of things like the endoplasmic reticulum, the Golgi, the mitochondria. All those organelles inside the cell are also encapsulated with a lipid membrane. So the essential oil molecules, because they're all lipophilic, they love lipids, they can permeate into and diffuse right into the cell and get inside. Other molecules that are water-loving, okay, what they call hydrophilic, they have a hard time getting in the cell because they can't get through the membrane. So they need some type of channel or special channel to get into the cell, a gateway. The essential oil molecules do not need a specific gateway to get in. They can just permeate and diffuse anywhere on the surface of the cell, right into the cell, and then they can get into any other compartment in the cell. And because of that lipophilicity of the molecules themselves, they pretty much have access to any biological component in your cells, making them highly potent, okay? So that's fact number 2. Now, fact number 3. Now, this is what really got my interest once this clicked for me and the light bulb went off. So my whole life, studying pharmaceutical science, we were taught monomolecular therapy. And they don't even call it this because they don't even have a name for it because it's all that exists. So they don't even give it a name. So this is a name that I came up with. So monomolecular therapy is really the idea that you find one molecule that you take as a pharmaceutical drug, and that one molecule then is interacting with one target in your cell, the target that either reduces the symptom or tries to help with the issue that you have. Okay, so that's monomolecular therapy, one drug. Polymolecular is when you have a mixture of different molecules, which is what essential oils are, or plant extracts are. That's actually a mixture of many different molecules in a one vial, right? And so you're not taking one molecule or one constituent, you're taking many. So most essential oils are going to be somewhere between 20 different constituents up to 300 different constituents. And that all depends on the growth of the plant, the harvesting of the plant, and the distillation process that's used, time, temperature, and pressure. That will determine how many of those constituents in the plant end up in the oil. Okay, so Young Living does a very good job of making sure that they get all of the constituents out of the plant so you get closer to that 300 number as possible. And in getting the trace constituents, things that are in there at very small concentrations, less than 0.1% of the total mix. All right, so there's some constituents that make up a large portion of the essential oil, like 60 or 70% of the oil may be this one constituent. And then the rest of the oil, the other 30 or 40%, maybe hundreds of different constituents. So they each have a different concentration in that oil. And that ratio of those different constituents is what gives the essential oil its therapeutic nature. So the polymolecular nature of essential oils and other plant extracts is what actually creates this environment where you have something completely different than the monomolecular approach to medicine, which is pharmaceutical science is all based on. So I'm going to explain how that works. So when you get inside the cell, if you're actually to zoom into a portion of the cell, people think of a cell like a bag of goo, like it's just like a bag of water or something like that. And there's not much structure going on in there. What we found as we started studying the cell more, molecular biologists, we found little nanomachines in there and interactions between them, and millions of different players, millions of different nanomachines, all working together in this integrated functional system that creates the cell. This is just an artist's depiction of what it may look like once you get in you zoom into a very small portion of the cell. So it's very busy. There's lots of things going on. And so it's not just like a bag of goo, okay? So what molecular biologists do is they look at all of the different proteins the cells make. So the proteins are what make the cell do what it does. So they carry out the function in the cell. The proteins are the biological molecules that do the work. DNA is just the instructions. Doesn't do any work. The proteins carry out those instructions, okay? Can everyone still hear me okay? Okay. So the proteins carry out those instructions. In the human genome, that all of the different proteins at the proteome that the human cells make is about 1 million different proteins that the human cell makes. And each one of those 1 million different proteins has a specific function. Some control blood pressure, some are receptors for your neurological system, so on and so forth. Okay, so each one of these proteins, they represent as a little dot, a colored dot in this network. Now, this is just a subset of them. Obviously, this is not 1 million dots, but this is a subset of the proteins that are in our cells. And these proteins interact with each other. They communicate, they bump into each other, and they bind to one another, and they communicate with one another. This is called a network or an interaction network. You can think of it like the World Wide Web where all the computers are connected together. Well, inside of our cell, these proteins interact with each other. And so biologists or molecular biologists represent that through this type of diagram called a cellular interaction network. And you're actually looking at which proteins this, like for example, this little blue protein here, which protein does it interact with? Well, it interacts with this guy right here. And then this one interacts with this one, and so on and so forth. And so from this type of diagram, you can predict if I mess with this protein, what are the downstream effects of that in the cell? Because I can follow the ripple effect through the cell and see where it interacts into the cell. That's why these types of network diagrams are valuable. And this is how sort of like drug discovery works, because they're making a drug against one of these proteins. For example, this red one right here, Pfizer or someone may design a drug that binds to that one protein. Again, that's monomolecular therapy. But then that's not going to be the end of the story, because once you interact with that protein, it's going to ripple throughout the cell through the cellular interaction network and perturb or change what's happening inside the cell. So to give you an idea, a very simple cartoon diagram, in Western medicine, you have one drug compound like this blue triangle here that's been designed to interact with one target, okay, like this orange ball. All right, so that's Western medicine, monomolecular therapy. But in polymolecular therapy, you have many different constituents, which are represented by these different shapes and colors. Okay, these are up to 300 different constituents that may be in an essential oil, right? And then here is the cell that is these molecules are going to go into. And then each one of these little tiny points is one of the proteins that these compounds or constituents from the essential oil are going to interact with. And so this is a much different idea than this. Can everyone see that? That you're going to get a much different effect if you have all of these different molecules. Now, the thing to realize is all of these different molecules are different shapes and sizes. They're not all the same. They have a diversity of chemical features to them so that they're not all alike. So they're all going to interact with different targets in this network. All right, so the 300 different constituents aren't going to interact with just one protein. Constituent number one may interact with this yellow protein. Constituent number two might interact with this red one and this blue one, so on and so forth. So you can see this gets really complex, but you can see that it's much more complex than the way pharmaceutical or Western medicine reduces it to one compound. So this is what the light bulb that went off in my brain when I started to study how this worked, because all I was taught was this. Okay, so for example, if you take ibuprofen, okay, this molecule ibuprofen interacts with one target, and the target it interacts with is called cyclooxygenase. All right, cyclooxygenase is a protein that can cause pain and inflammation and fever and all of that. So if you block it with one drug, then you can relieve those symptoms. Okay, so that's how monomolecular therapy works. If you take a blood pressure medicine, if you take a cancer medicine, if you take any type of medicine, it's one compound designed to interact with one very specific target. And that's how it all works. All right, so that's fact number 3. Fact number 4 are essential molecules are generally safe. So drug compounds, because they only interact with one target, that tends to really perturb or disturb the cellular interaction network and cause it to shift off of equilibrium. Okay, so you're only pushing on one part of the network. So this can cause a lot of side effects. Okay, essential oil molecules, because there's so many of them, there's synergistic interactions. With synergy, we have 2 or more things happening at one time. And then you also have quenching, where 2 molecules are basically working against each other. And this can be a good thing. You might say, well, that sounds bad. Well, quenching can actually stop a side effect from happening. Monomolecular therapy with drugs, you don't have that quenching. You don't have another molecule to block some side effect. So I'll show you what I mean by that. So essential oil molecules are safe because they have these synergies and quenching interactions that basically even out the response in your cell so that you don't have these crazy side effects. So it makes them generally safe. So to give you an example of this, okay, if you look here, here's one molecule from a drug. This is I'm looking at right now is the Western approach. Here's the drug compound you take into your body. Okay, it interacts with this blue protein in the network. All right, now this blue protein communicates with this orange, yellow, and green protein, right? The network, and then these proteins interact with these, and then those interact with these, so on and so forth, and then they all converge on this red protein. Now, when you take this drug molecule, interacts with the blue one, it's designed to do this and then give you some positive effect or reduction of some symptom, right? So that's the positive aspect of the drug molecule, you're basically reducing some symptom. So that's its effect right here, what I'm showing through this interaction, okay? But there's unintended effects here because remember, you got a network all connected together. You can't stop this flow or this ripple effect from occurring. So all these other other proteins are signaling each other once this one binds, and it just ripples down until this red guy here gets activated, and now you have some negative effect or a side effect that you did not intend for the drug or want the drug to have, but there's no way around it. You have to have this negative effect that goes with this positive effect, right? Because it's the way the network works. But if you have polymolecular therapy, which is this slide, and here I'm making it very simple. I'm only looking at 2 compounds instead of 1. So this is the first example of polymolecular therapy. We're just using 2 drug compounds instead of 1 drug compound or 1 constituent. So you have this molecule here interacting with the blue protein, and you have this other molecule, number 2 here, interacting with this yellow protein. Okay, so it's the same deal. This blue one gets interacted with. You get the positive effect like we had before, and it ripples down. Okay, but this second molecule basically binds to this protein, this yellow one, and quenches that signal from being passed along to the green guy. So remember over here, if I go back a slide, you see that signal got past the yellow one, then the yellow one passed it to the green one, and then finally to the red one. And this red one actually needed all 3 signals here to get the negative effect. But in this case, because we blocked one of the signals by quenching the yellow protein, this red protein down at the bottom no longer gets all 3 signals, it only gets 2. It's missing this one now. And now you don't have that negative effect anymore. You don't have that side effect. So that's called quenching. And this happens all of the time with essential oil molecules or essential oils, where you have other constituents in oil that quench the negative effects of one of the other compounds. And this is very important, because if you get into where people say, hey, essential oils are not safe, right? They're dangerous. And all of those types of statements are made on the premise that some scientists purified one of the constituents from the oil. They basically reduced it back down to monomolecular therapy. So for example, they took lemon oil and they purified one of the constituents called limonene. And then they took the limonene and they gave it to rats or something, and they noticed that the rats developed tumors when you gave them very high concentrations of limonene. And now all of a sudden everyone's screaming, oh, essential oils cause cancer because we saw limonene do this in a rat. That will always happen when you take one compound, okay, away from the rest. So the way this has been designed and engineered into nature is that the plant compounds work together as a team in these synergistic combinations and quenching interactions so that they ameliorate and help one another so that there's not a negative effect in the body. Now, that's not to say that you can't overdo it with essential oils. You have to be safe with them, because anything can be dangerous if you take too much of it, even a natural product. You can basically drink water too much and die from that. Water is normally good for you. But, it's like that with every molecule. It doesn't matter if it's generally safe. If you take too much of it, your body can't deal with it. So there are safety issues you have to deal with essential oils, but they're nowhere near the orders of magnitude different between essential oils and the side effects that they have and the concentrations you have to worry about, and drug molecules. With drugs, it's very easy to overdose, and very small concentrations can actually lead to very potent side effects. This is not true with essential oils, and it's mainly because of this quenching that I'm talking about. So you can't really judge the safety of an oil by individual constituent monomolecular type safety studies that are done on the individual constituents. Okay, so fact number 5 is synergy. So essential oils, molecules, plant-derived extracts exhibit synergy. Synergy, what is that? There's different types of synergy. So in my book, I actually talk about 4 different types of synergy and how they work. I'm going to talk about the most common one tonight, which is synergy. What most people think of is you have, multiple compounds, and you look at just any one of them, you have a certain effect, bioactivity to that one compound. But when you put two of them together, the total bioactivity of those two is greater than the sum of the two individual components by themselves. So for example, 1 plus 1 instead of equaling 2, 1 plus 1 might equal 4 when you're talking about molecular compounds. And I'm going to show you how that works. Now, with polymolecular therapy, that's not true at all because, with monomolecular therapy, that's not true at all because you don't have more than one compound, so you can't have synergy by definition. So with Western medicine, you can't have synergy. It's impossible because it's only one compound. Essential oils have this. Now, let me show you how it works. And once you see this diagram, like light bulb goes off in your head of how this works now. All right, so this is a very simple model, but it helps you understand and visualize how synergy works. So on the left here, I have scenario 1, where you have just one molecule. Again, it's monomolecular therapy. And you have the target that this molecule is designed to interact with. But if you look here, there's a door that's shut, this green door, where the molecule could not access the target. So if you take this molecule, it doesn't matter how much you take of it, it's never going to have any bioactivity because it can't get through the door. It doesn't have the key to the door, it can't get through the door. So basically you have zero activity. So you can take as much of this compound as you want, it's still zero is the end result. But on the right, I have scenario number 2, which is, can everyone still hear me? Okay. So on the right, I have scenario number 2, where we have two molecules. This molecule here, this constituent, can open the door. It has the key to the door, so it can open the door. The second molecule, which is this first one here, now gets through the door and can interact with the target right here, Pac-Man, and now you have the bioactivity, okay, that you want. Now notice, no matter, you can take a very small amount of molecule number 2. You don't need a lot of it because all it has to do is open the door, right? Once the door is open, lots of molecule 1s can flow through this door. Hundreds, millions of them can come through now, interact with millions of targets. But you only need one of these molecules to open the door. This is synergy because on the left, you had no activity with the one molecule. Now notice if you just had molecule 2 by itself, it would open the door, but then there wouldn't be the first molecule to come and interact with the target. So you'd still have no activity, right? So molecule 2 by itself gives you zero activity, right? But if you put both of them together here on the right, now you have some kind of activity. They have to work together. One without the other is useless. And so you need both. But going back to what I was saying, you don't need a lot of molecule 2. You can have just one, and it will open the door and allow lots of molecule 1 to come through. So molecule 2, you can think of this like the trace constituents in the essential oil, things that may be at 0.01%. The constituents that other essential oil companies don't worry about are these keys that open the door. And they make the difference between having therapeutic activity and not having therapeutic activity. Because if you're missing the key, it doesn't matter if you have 70% of your oil made up of pinene, okay, one of the other constituents, or limonene. It doesn't matter if you don't have the one trace constituent. And the only way you get that trace constituent is if you do the right distillation processes and you do the right types of chromatography and have the right software and compound libraries in your analytical lab to show that you have that trace constituent present. So that's the power of synergy. And this happens incredibly in many different instances. This is just a very simple model of 2, but can you imagine the amount of synergy that's taking place when you have 300 different constituents in an oil? All right, so that was fact number 5. How am I doing on time? Let's see. I don't know if I can see the clock here.
You have about 25 minutes.
Okay, I'm still doing pretty good. Okay. So fact number 6. So drugs deal with symptoms of a problem, but essential oils do something completely different. They deal with homeostasis or equilibrium in a cell. Okay, I'll show you what I mean by that. So you're sick, you have some symptom. Okay, you experienced the symptom. So let's say your car doesn't have oil in it. All right, that's the problem. You don't have oil, but your light on your engine comes on. The oil light. So is the oil light coming on the problem, or is it the oil missing that's the problem? Well, it's the oil that's missing that's a problem, but the oil light coming on is indicating that there's a problem. So the symptom is indicating there's a problem, but it's not the problem itself. Just like the light on your dashboard is not the problem. There's the way the cellular interaction network works, and you saw how complex it is. All these different proteins interacting with each other. They can all converge at the same symptom, these different pathways in the cell, and basically make you have the same symptom, but you don't know which pathway is causing the symptom. You don't know the root cause that's leading up to the symptom. So here I make it very simple. You can have pathway number 1 in the cell giving you the same symptom as pathway number 2, path 3, path 4, path 5. There's something wrong in one of these pathways, but it's causing the symptom. So it's actually more complex than this, but I'm just making it simplified for you. So how do you know which symptom to work on? Which pathway to work on? How do you know which pathway to fix if you just have the symptom to look at? Okay. So with drug companies, they have to spend $800 million on research and development for 13 years on average to bring one drug compound to phase 1, 2, 3 clinical trials, get an FDA approval. That's 13 years, $800 million. They can't afford to develop a drug compound for every single one of these pathways. So they can't do it. They can't predict which pathway you have compared to someone else's. That would be too individualized. I want to give everyone the same drug compound. The only way to do that is to deal with the symptom. It's the only practical way to do it. So that's why all the pharmaceutical companies, all the drugs you take, the monomolecular therapy, is because the only thing they have to do is block the symptom. That's right here. So they develop a drug and it blocks the symptom because they can't really get at the pathways. It's too formidable of a challenge. It's just impractical. It will cost them trillions of dollars to do it. So they don't know which pathway to fix. They just assume one of these pathways is broken, but we're going to just deal with the symptom. So it's like ibuprofen, again, cyclooxygenase. That's really just a symptom here. You're blocking the symptom, but you're not dealing with the cause of the problem to begin with. Now, when you look at plant constituents, you're not really coming in here and just blocking the symptom. You actually have constituents, because you have so many of them interacting in the cellular interaction network, you're basically modulating or affecting each one of the different pathways. And each of the constituents is basically, you think of hundreds of interactions or thousands of interactions inside the cell, and they're each basically gently nudging one little aspect of the path of the cellular interaction network and just basically pushing it back into equilibrium, little tiny individual but summed up little nudges. Okay, so these constituents are basically creating an equilibrium, restoring homeostasis back to each pathway, right? And so now that's how you're dealing. You're kind of getting at the root cause of what's going on. You're restoring homeostasis or equilibrium back to the cell, rather than just trying to block the symptom, which is what pharmaceutical medicine does. So it modulates, the molecules themselves modulate multiple pathways to restore some kind of equilibrium homeostasis. So you're getting at the root cause of the problem here, rather than just trying to block the symptom. And because of that, when you use essential oils, you have more of a probability, a higher probability of actually finding a solution to your problem, right? Because you don't know, you're not going to know either. Maybe, You have pathway number 5 here messed up, giving you the symptom, and somebody else has pathway number 2, right? But you don't know which one it is. So how do you know what to do? Well, when you take essential oils and then you start blending essential oils and you get even more constituents, right? From blending them, now you can start to interact with more pathways, all right? And you increase the probability of finding a solution to your problem. So that's fact number 7. All right, so now I'm going to get into some of the secrets that most people don't know about pharmaceutical medicine. And it's kind of embarrassing for them, actually, because they like to hide this. But multiple studies have shown that greater than 50%, actually 52%, of the drugs that are out on the market from the FDA that have been approved by the FDA, 52% of them were derived from some natural compound found in nature, either directly copied from a molecule found in nature or initially inspired by a molecule found in nature and then modified or derivatized into a different form to have higher, bioavailability or whatever. So that's a huge secret in the pharmaceutical industry, and I'm going to show you some examples. Now, in the 1800s is really when they started isolating. Instead of just looking at plant extracts, they started the monomolecular thing, where they started just extracting one compound out of plants. So in the 1800s, all these drugs they had in the 1800s, in the 19th century, and some of the drugs they still have today that are based on this, all came from a plant. So here are some examples: morphine, codeine, atropine, caffeine, cocaine, ephedrine, pilocarpine, physostigmine, quinine, salicin, theobromine, theophylline, tuberculinine. So all of these are been pharmaceutical drugs, and some of them still are, that were extracted from a plant. Okay, even like aspirin, it comes from the bark of a willow tree. Okay, acetylsalicylic acid, that comes from the bark of a willow tree. So these drugs were all extracted from plants and purified, but again, they were extracted and purified away from the other natural constituents that were found with them. So they basically purified it and reduced it to a monomolecular type situation. So why do pharmaceutical companies go after, let's see. So here's a study that was done. So they looked at all the drug compounds. They looked at 1,186 molecules that were approved by the FDA from 1981 to 2006. And out of those, 52% of them were directly derived or inspired by a natural product. Okay. Why is that? Well, when you actually look at the molecules found in plants, they're very diverse and novel. And so what I mean by that, if you think of measuring some chemical feature about a specific molecule and you graph it, okay, so I'm not going to explain what each one of these things are, but just imagine a three-dimensional graph where you're measuring 3 different properties about each molecule, all right? And so you're representing it at this 3-dimensional graph. So the green triangles are individual molecules found in nature in a plant somewhere. And the black diamonds right in here are the molecules that that pharmaceutical companies or chemists can actually synthesize and invent and synthesize in the lab, an artificial molecule. And the green ones are the natural ones found in nature. So what you notice here, they're actually plotting this for actual compounds. And you can see that the natural products have a greater diversity in 3-dimensional space than the artificially derived compounds made by man. All the black ones are clustering right here. So they're all the same. They're very similar in nature. There's not a lot of diversity. There's not a lot of novelty. There's not a lot of difference between them because they're invented by man. But here, these compounds, the ones found in nature, are highly diverse in structure and in function. And in fact, they have a structure and function and novelty and diversity to them that can't be replicated by synthetic means in the lab. Even if we wanted to synthesize some of these molecules, we can't, because the molecular structure is too complex. We can't devise a chemical synthetic process, no matter how many steps we put into that process, to go from molecule A to molecule B and end up with the final plant compound, because the molecules, most of them are just too complex for us to do that. Okay. So here's a quote by Scripps Research Institute in Florida. Natural products remain the best sources of drugs and drug leads, and this remains true today, despite the fact that many pharmaceutical companies have de-emphasized natural products research in favor of high-throughput screening of combinatorial libraries, these are artificial libraries I was telling you about, during the past 2 decades. Natural products possess enormous structural and chemical diversity that is unsurpassed by any synthetic library. About 40% of the chemical scaffolds found in natural products are absent in today's medicinal chemistry repertoire. Natural products represent the richest source of novel molecular scaffolds in chemistry. That's basically what this graph is showing you. And pharmaceutical scientists and everything realize that, recognize that, so they always go to nature to get inspiration for their drug design. That's where they've had the greatest success. Dr. David Newman, who was part of the National Cancer Institute of the NIH, he headed up the division called the Natural Products Branch. He's published dozens and dozens of papers about this topic, so he's very well known for it. And here's one of his quotes. Chemists started making libraries of hundreds of thousands to millions of compounds, but they were simple compounds. Mother Nature doesn't make simple compounds. Mother Nature wants compounds that fit into particular places. So the proteins in your body have a specific three-dimensional shape to them. Each one of the million different proteins I was telling you about earlier has this very specific three-dimensional shape. Just to give you an idea, this is one protein in your cell, and look at the complex three-dimensional shape of that one protein. Now, there's a million different of these proteins, and they all have a different shape. All right, so you need different structural shapes to interact with these proteins, and plants are very adept at producing molecules that can interact with the highly complex structures found in the proteins in our cells. All right, so this is what a drug company has to do to make a new drug. So they start out here on the left with millions of compounds. They filter it down to tens of thousands. Then they get it down to 250 or somewhere in that region. Then they hopefully in clinical trials, they have 5 or less, and then FDA review, and then 1 approved drug. Again, like I said earlier, this process usually takes about 13 years on average, and it costs approximately $800 million. So that leaves them with 7 years on the patent, because they patent back here, okay? And then they have a 20-year lifetime on the patent. By the time they get it through FDA and marketing and manufacturing, they usually have about 7 years left. In 7 years, they got to make up the $800 million they just spent on research and development. So they have to charge a lot for the drug. They need $1 billion drug. They need $1 billion blockbuster to pay for all this. That's why drugs are so expensive. But what we used to do in the company that I started is we helped them find compounds faster by developing tools that allowed them to do that quicker. And this is sort of like how they do this. They do this with robotics. They have these little these dishes here with little wells in them. This is a 384 well plate. And in each one of these little tiny wells in here, there's the drug target, what you're trying to bind to. And then in each one of these pipettes, what they're squirting into those wells is one of their molecules in their library. And they're just screening through these with robots all day long. And a high throughput drug screening library kind of looks like this. Okay, and these robots are just screening the millions of compounds they have, looking for a hit against the target, monomolecular therapy. But again, a lot of these compounds that they have in their library are inspired off of plant molecules. So that's one way to do it. The other way is called rational drug design, where you're not screening a shotgun approach and hopefully one fits. In rational drug design, you actually start and you try to design a molecule from scratch using computer tools and simulation tools or plant or compounds from nature, and trying to come up with a design instead of screening millions of compounds. We'll skip that. Okay, so I'm going to start to summarize here. So some of the things we learned is that essential oil, about why essential oil molecules are so useful and powerful in how they work, is first, they're small, right? We saw that they're about the same size as some of these pharmaceutical drugs, but they're much, much smaller than a cell, about 50,000 times smaller than one of our cells. And among the biological molecules found in our cells, essential oil molecules are right there with the smallest molecules. All right. This helps them to get to any location in the cell and interact with all the little nooks and crannies inside of these complex three-dimensional shapes. Essential oils molecules are lipophilic, meaning they like fat molecules. They like to dissolve into fat molecules, which is the membrane of our cells, the membrane of the nucleus of the cell, and other organelles. So they can basically transport themselves anywhere they want in the cell without needing a gateway to get in. We learned that with the Western approach to medicine, it's basically built on one molecule, which we call monomolecular therapy. And with plant extracts, this is polymolecular therapy instead of monomolecular therapy. All right, which is many, many compounds working together in parallel with one another. We learned that drug compounds, because it's monomolecular, really perturbs the cellular interaction network, skews it in one direction, because you just have one interaction point. Now you're taking it out of equilibrium, and through that downstream ripple effect through the network, you get side effects. And these come along for the ride with drugs. There's no way around them. So all drugs have a litany of side effects, and usually at very low concentrations. Because essential oils and other plant extracts have this polymolecular approach, there's synergy, there's quenching that goes on in the network, and this kind of evens everything out so you don't have these very harsh side effects. But through these synergistic interactions, which again are only possible with essential oils or plant extracts, are not possible with drug medicine, you cannot have synergy. So you can never benefit from synergy with Western medicine. But essential oils, you can do all types of different types. Even in one oil, a single oil by itself already has synergy occurring from the relationship of the different constituents interacting with the network. And I showed you the simple model with the door opening, and you needed two molecules, one to open the door and the one to go through, but they both had to work together for that to occur. So that's happening even in one single oil. But when you start blending oils together, then you can get even more diverse and novel types of synergy taking place. Again, we learned that with the synergy, this trace constituents in oil are extremely important. With Western medicine people think algebraically. They think like if you take a plant extract, and they would say, okay, this plant extract has 80% of constituent A in it, and this plant extract helps with whatever. So they would say 80% of the potency of this plant extract is coming from this one constituent, and they would purify it away. from the rest. And so they think algebraically, they don't realize that there's another constituent in there that actually is activating the power of the other one. And both are needed. And so it's not algebraic. So something might be something, a constituent might be in the oil at 0.001%. And that makes all the difference in the world. So you just can't look at the percentages, right? Biological systems are nonlinear. You just can't look at it like an algebraic sum. This is how synergy works. 0 plus 0 can equal 5 with synergy, right? Something has 0 activity, another compound has 0, you put them together, now you have 5. So that's not algebra. So this is nonlinear. So this is how biological systems work, and this is the power of polymolecular therapy. You can't have that with monomolecular. Let's see. Okay, so homeostasis, which is equilibrium, because you have so many different constituents interacting with the different pathways in the cell, each of the constituents are basically nudging or pushing the network back to equilibrium where it's supposed to be. This is homeostasis. And that's why it sort of has a gentle effect to it, right? So there's not these harsh side effects, but it's a very gentle approach to restoring health. Because symptoms are basically the only thing pharmaceutical companies can deal with because of the model I just showed you of how they do their research and the monomolecular approach. They can't afford to go after every pathway that's the root causes. They can only go after the symptom. They develop a blocker that blocks the symptom. That's what they do. And that's it. So you're just blocking the symptom. You're never dealing with the root cause of your issue when you take a pharmaceutical drug. I don't know of any examples of a pharmaceutical drug that we're actually dealing with the root cause of the issue. They're all blocking some downstream symptom after the root cause has already occurred, downstream from the root cause. So they can't go after the root cause. They can only go after the symptom. But when you get into polymolecular therapy, because you have all of these different possibilities and interactions with the constituents in the network, now you can start to affect or modulate all the different possibilities of root causes. And now you have a much higher probability of restoring your health, but also dealing with the actual problem itself. Big secret that they don't want you to know, that most of the drug compounds that are out there were first designed from plant compounds, and then this morphed into the 20th century where they just use a computer or something else, but they're basically copying molecules found in nature. Because why? The natural compounds have a very high chemical diversity and novelty and complexity that can't be replicated by artificial synthetic means in the lab, and because of that, they just can't mimic it 100%. So they have to use these compounds as inspiration to design drugs. And many of the drugs that most people use in their cabinets, and these people will mock you for using essential oils as some oil quack. I get mocked all the time on Twitter, being an oil quack doctor. But, you'll get mocked, and these people are going to their drug cabinet and taking a pharmaceutical drug that came out of a plant. They don't even know it, and they're mocking you for using essential oils. And it's this chemical diversity inside the constituents that gives them their power, the superiority over man-made compounds. So that's the end. I'm going to stop there because I'm probably out of time. And hopefully that was useful to you. Again, this is really module 2 of my 7-module series. You can get the entire series at starfishsense.com. And if you get any 2 or more items tonight, you get 30% off. And you got the book if you want to read it. A lot of what I've said tonight is in this book, and I go over it in more depth. And there's application methods in here and how to use the oils and how to pick an oil, choose an oil, how to blend oil. All that is in this book as well, so you can get that. And I make it very simple, like I did tonight. I try to make things on a conceptual level so it's not too complex. And then the memory stick has all my stuff on it if you want to get that. And that's it. And I don't know how much time is left, but if there's questions or anything.
We do have a few questions, son.
Okay.
So Joan asked, she said, questions for Doug later. So please explain further how about how receptor sites work. Do you have a quick answer on that one?
Receptor sites. So on the outside of every cell, there are protein receptors. There's receptors for everything, right? So there's receptors for signaling, for neurochemistry, for hormone signaling. So cells communicate with one another by secreting some molecule, and that molecule travels and then interacts with the receptor, and then the receptor binds to it, and then a signal is transferred inside the cell from that receptor that instructs the cell what to do. Essential oil molecules do interact with receptors. To give you an example of an essential oil molecule interacting with a receptor, beta caryophyllene, BCP, which is found in copaiba, interacts with the cannabinoid 2 receptor, which is a receptor found in mostly immune cells and as well as neural cells or the nervous system. But yes, there's many examples of essential oil molecules. And now you can interacting with receptors. Now, a molecule can actually block a receptor. It can activate a receptor. It can modulate a receptor. It can turn the volume down of a receptor. It can competitively inhibit. It can allosterically modulate. There's different ways that molecules can interact with the receptors, not just one type of interaction. And essential oil molecules are known to do all of the above in terms of interacting with receptors, whether they block them or activate them or inhibit them. So there's different interactions there. I don't know if that answers your question, but that's sort of a general answer to your question.
Could you stop sharing your screen real quick so we can see you better?
Yeah.
And here's the next question. I know part of the answer, but I'll let you answer half of it. Carolyn asks, since a lot of drugs are taken internally, can we now see that effectiveness of essential oils are versus drugs? And what would you say is the most effective way to use the oils, topical, inhalation, or ingestion? I know because I just studied your material that testicular use of oils is very good, but we all don't have testicles. So how is the other way we can use this the best?
So the question is the different application methods.
Yeah, which's the best way to apply the oils to be able to absorb them internally? Topical,
Okay.
inhalation, or ingestion?
Okay, so in my book I go over this in one of the chapters. I also have a class that goes over this called essential oil application methods. But in general, if you do, can you excuse me 1 second? Get my dog out the door there. So many people don't know this. If you ingest an oil, the first thing it does is it goes to your liver through the hepatic portal vein. And in your liver, there's phase 1 and phase 2 metabolic chemical reactions that take place. And those metabolic reactions actually alter the molecules somewhat. They derivatize the essential oil constituents and change the structure of the molecule somewhat. And this then the molecules enter into your bloodstream and travel throughout your body. Okay. So, but when you apply essential oil topically, that doesn't happen until eventually circulates through your system and your liver gets a hold of it. Then it modulates them, or changes the molecules. But you have that initial, when you apply them topically, you have that initial traveling throughout your body so that the molecules in their native form interact with the cells in your body before they get modified by your liver. But when you ingest an oil, the liver modifies them before the cells in your body see them. And so there's a different bioactivity profile for essential oils if you ingest them as opposed to topically applying them. But that's not the case when you're talking about your gut. So when you ingest the oil, your gut, your intestinal lining and stuff is going to see the oils in their native form because they haven't been modified yet. And then when they get through to your liver is going to see them in their native form as well. But then once they leave your liver, everything else is going to see molecules that have been metabolized to a certain extent so what your liver is trying to do at that point Your liver is trying to excrete these molecules out of your body. They want to get the molecules into a form. Remember I said essential oil molecules are lipophilic. They like lipids. So that's not in a very good form for you to pee out, because for you to pee out something, it's got to be water soluble. So if your body wants to get rid of this molecule, it's got to be able to pee it out. So it's got to be more water soluble. So your liver is actually modifying the essential oil molecules to make them more water soluble at that point, so that you can actually pee them out. Now your kidneys are going to do that as well. They're going to actually do some more metabolic processing on the molecules to change their structure before you pee them out. So that's what your liver is trying to do once the molecules enter into your body. So you're going to have a different profile. Now, in terms of speed of how fast the molecules get in your body, breathing in the oil is the fastest. Like within 10 seconds, your lungs, the molecules permeate your lung tissue. Is very thin. The epithelial layer in your lung, very thin, gets right into your bloodstream almost immediately as soon as you inhale them. The problem is, and there's no metabolic processing, but the problem is you don't have a lot of concentration there. It's very diluted out, parts per million. So you're not getting any kind of real dose in when you inhale oil into your lungs, into your bloodstream to affect your body. The benefit of inhaling the oils is the olfactory response and activating the receptors, the 350 different olfactory receptors in your olfactory bulb, and then go into your limbic system. So that's the most benefit from inhaling an oil or trying to help your lungs with something. But really getting the oils into your molecules into your bloodstream to say affect your kidneys or something, you don't want to, inhaling, you don't get enough of a dose. But the next fastest route after inhaling would be topical. Topical, on average, it's about like 20 minutes, but some can be 2 or 3 hours before the molecules get into your bloodstream, based on their size and shape and structure, depending on which constituent you're talking about. Not all of them enter into your bloodstream in 20 minutes, like you see on these memes that go around. Some of them take hours, but that's sort of the second. And you have a very large dosing window. You can go very low on dosing, higher on dosing, just depending on how much you dilute the oil and how much you apply to your skin and the surface area that you use. You have a very large dosing window, and they're not modified. They're not metabolically processed. But when you ingest an oil, now you're going to have that metabolic processing. But so I usually tell people to ingest oils when you're talking about something maybe as a GI issue or a liver issue, or maybe even a urinary issue. I would ingest. But if you're doing, if you want to do something with your brain or something with your cardiovascular system, then or some other organ or system of your body, skeletal, muscle, nervous, I wouldn't use ingestion as the major avenue. So I hope that helps.
Thank you. That was quite a short answer. So,
Was that
okay. There's so many wonderful comments. Someone said, I don't usually like science. This was Jennifer, but this was awesome. And thank you, Dr. Doug. It was super informative and interesting. Michelle said, best call I've attended, phenomenal call. And there's a whole bunch of people who want to get your information and they want to know if you have a special discount code.
Well, the discount I have is if I don't have a special code, but if you're getting 2 more tonight,
It will automatically do the discount in the card.
it automatically apply to 30%.
Okay.
And if you get the memory stick, it's if you add up all the courses and everything on the memory stick, it's like almost twice as much than just getting the memory stick. So there's a discount there.
Yeah.
And if you get the memory stick plus something else, and you get another 30%.
I didn't want to wait for the memory stick, so I just bought it online a couple of weeks ago, and so he'll send you a digital link or the memory stick if that serves you best. But phenomenal information, and I don't think I got your book on whatever you said with the light. What was the name of the light one? The biblical one.
Are you guys still there? Okay,
Yeah.
I didn't see you. Like you went away for a second. It's called The Author of Light.
The Author of Light. Yeah, I'm
Author
gonna
of
get
Light. Yeah.
Right. Okay, perfect. Doug, thank you so much. I know that we would all love to have you on again, and we will, and as long as you say yes, and so that we can get more of this
Yeah.
information. He has phenomenal courses, as we just said, and even if you bought them all and watched them all, you'd still want to hear them again. So thank you so much. And we just thank you for all your time and all your expertise and knowledge and willing to share. And thank you so much to your wife and whoever introduced Amy to the oils. Otherwise, we'd be sitting here in the dark, right? So I appreciate all that. And right now I'm going to stop the recording so we can get ready to go to the next caller.